Good morning. Today we are going to start a new topic, water microbiology. In water microbiology, we will study the microbiota of water, and we know different microorganisms is present in the water, like bacteria, fungi, their spores, even though many type of algae and many type of microflora also present. So these are all combinedly we are calling water microbiology, and we are taking only those organisms which we can see by the microscope only. So those microbiology is the study of microorganisms and their communities in water environment. And another term we are calling aquatic microbiology. So aquatic means water, while water microbiology relates to the study of microorganism in potable water. So potable water means the drinking water, the water we can able to drink it. So the scope of aquatic microbiology is wide and include the habitat like plankton, benthos, microbial mates, and biofilm, which may be found in lakes, rivers, streams, seas, groundwater, rain, snow, and hair. And these type of different water is there, and they are all having different type of microorganisms. And these microorganisms we are dividing into the planktons, benthos, microbial mates, and biofilm producing bacteria. So these microorganisms we will discuss one by one, and we will separately have section in this topic. Now, drinking water microbiology. So we talk about the potable water, and before going further, we have to know what characteristic is there of potable water. So potable water is a water that is free from pathogens and chemicals. So no type of contamination should be there, and WHO and NIRI in India, they gave the potable water standards, and these standard all over the India and WHO standard all over the world they are following. And when we are calling the potable water, it means the water we can able to drink, and it is for free from the all type of pathogens and chemicals. These pathogens and chemicals we are countering those pathogens and chemicals which is dangerous to the human health, but pathogens which is the dangerous to the human, and chemicals also. So they are it is present in the water, and so that water is not potable. So any taste, odor, and color must be absent from the potable water. Then only you can able to drink. Otherwise, if taste is different, odor and color is there, then you can't able to drink that water. Then we are not considering that is a potable water. So raw water may contain many contaminants and dried from the sewage and nearby industries. And these contaminants we can categorize in the chemical contamination or microbial contamination. And these contamination may be coming from the sewage water. Sewage water is mixing with the your source of drinking water, and the nearby industries also they are effluenting the sewage in the potable water source. So many enteric type of pathogens are waterborne, and they are the many disease causing bacteria, and these disease causing bacteria we are drinking knowingly and unknowingly, because the potable water is very less if you will see in the on the earth, and these potable water is available only in some areas, and every person on the earth is not having accessible to this potable water. That's why the enteric pathogens are causing many waterborne disease, and these disease are the many positive as cause of the many deaths of all around the world. So therefore, water is treated and disinfected before using. So whenever we are using any water from the any water source, before that we have to treat with the disinfectant, and then we can able to utilize it when we will confirm that there is no any pathogenic bacteria or microorganism is there, or any type of chemical is not present. So the water should be safe before drinking, and we have to confirm it when we are utilizing or when we are taking water. Now, uses of water bacteriology. So, water bacteriology is the study of the bacteria present in the water. Maybe it is from the river water or pond water or lake water. So, how we can use the water bacteriology, and when we are determining that whether a water supply is fecally polluted, and it is to test for the presence of normal fecal organism. So if the water bodies is mixed with the sewage water, so that they are having fecal organism, these fecal organism can able to cause uh, many type of disease. So before drinking, before if we are using those type of water, and if we will drink it without any testing, so that it may cause a dangerous type of disease. And before using, we have to check it whether it is polluted or polluted type of, or there is presence of fecal organism. So testing found normal fecal organism as indicator of fecal pollution, and it is a reliable way of determining whether water is bacteriologically safe to drink or not. So before drinking, we have to test it whether the any potable water, when we are thinking that it is potable water, and we have to test it so that we can determine whether water is contaminated or not. So if it is not contaminated any time by means of the microbiological and chemical, then it is safe to drink. So a single laboratory examination of any water does not justify the conclusion that the supply is safe for drinking. So bacteriological analysis of water should be performed at regular intervals. That's why the water supply department of government of Rajasthan and government of India, other states, they are having regularly checkup of bacteriological analysis, even though chemical analysis of the water. And when they are having the positive test of any fecal organism or chemical contamination, then they are removing those type of contamination by treating with the chemical and physical treatment, so that the water can able to change into the potable water. So these microbiological contamination and chemical contamination, we can able to remove it from the water bodies, and then we can able to utilize those water for the potable purpose. So an important aspect of water microbiology is to numerous disease causing microorganisms spread throughout the water. So the main problem with the water microbiology, the many pathogenic microorganisms is there, and they are the main causing of the many waterborne disease. And many bacteria, viruses, fungi, protozoa, they are responsible for waterborne disease. So the recurrence of waterborne illness has led to the improvement in water purification. And this water purification, many techniques we developed since the waterborne disease is the main problem of the world. And nowadays the waterborne disease is decreasing, and since we develop so many purification techniques. Now some terminology we utilize in this topic, and we should know these terminology before going further. So first one is potable. Potable, the term potable means clean and safe. So when the water is clean, then it is safe for the. So when we are talking about the potable water, it's mean the water is free from the any type of pathogens, taste, odor, color, toxin, radioactive material, organism, oil, gases, and any other type of extra thing which is not part of clean water. So these are all, if it is not present in the water, then we are calling it is potable water. Then water can be suitable for the drinking purpose. Next term is pollutant water. If these are all thing is present, only one thing is present means some microorganism is present, only another thing is not present, or mixture of two things like organism and oil is present, organism or odor is there, or color is there, or any type of toxins is there, two or more than two or single thing is there, then water is calling pollutant water. The source of the pollutant, it may be a sewage or it may be a industries. Okay, now sewage. So the community waste or garbage we are calling sewage. The community which is throwing the waste material and garbage and polluted water inside the river, pond, the lakes, and these sewage is contaminating the our potable water or natural source of water. So the source of the waste and garbage is our community only. So sewage term is utilized for the waste and garbage. Now, typical water quality standards. These water quality standard will show that the whether water is drinkable or not, or potable or not. So, drinking water standard is there, and we have to check before the drinking. So, drinking water should not contain any type of coliform contamination. Okay, zero coliform organism should be there in the potable water. Then recreational water is 200 fecal coliform per 100 ml is allowed. If less than is there, okay. If more than of that is there, then it is not okay. Then if you talk about fish and wildlife habitat type of water, so 5,000 fecal coliform per 100 ml of water is allowed, and if you see the more than that, it is not okay. Then shellfish type of water, so 14 fecal coliform per 100 ml is allowed. So these different type of concentration is allowed, and if more than of that parameter the concentration will go, then it is not allowing to, then we are not considering as a potable water. Now some organism, if it is present in the water, then we are calling it is an indicator organism. Now general coliforms it is present, so we are calling the indicator organism, like water is contact with the plants or animal, and universally they are present. Then fecal coliform is universally is present when the mammals and birds is directly they are in the contact with the water. So mammals and fecal bodies it will be there, and the water when it is in the contact with them, then it will be natural. So Enterococcus bacteria, it is a type of fecal streptococci bacteria, and the fecal from the warm-blooded animal when they are releasing these fecal bodies in the water. So these type of Enterococcus bacteria, it will be they present in the water, and if the concentration is too high in the water, so water is not drinkable or potable. Now, what kind of water is inhibited by the microorganism? So, the underground water, like mineral and thermal spring and groundwater, due to their oligotrophic characters, are usually habited by the sparse microflora that is represented by a low number of species with almost a complete lack of higher plant or animals. So surface water such as stream, river, lake and sea water are inhabited by a diverse flora and fauna. Microorganisms in those water are largely varied group next to the typical water species. Other microorganisms from soil habitats and sewage derived from living and industrial pollution. It may be there. So different water source is there. Underground water source is there, and surface water source is there. So these water source can be contaminated. Underground water source also can be microbiology and chemically contaminated, and the surface water they are more prone to get contamination because they are openly available and openly available to any other type of organism. So the surface water which is having more contamination, and the underground water if they are contaminating by the chemicals or releasing the sewage in the underground water. When sewage is mixed with the underground water, it may have chance that the underground water is contaminated. Now group of water organism. So now we will come up with the type of water microbiology, and microorganism occupy surface water in all of the zone. They may be suspended in water, plankton, and cover stationary underwater object, plants like that. So periphyton or live in the bottom sediment, benthos. So here three type of different organism is there. The suspended organism we are calling plankton, and the stationary underwater object or plants we are calling periphyton, and the organism which is living in the sediments we are calling benthos. Now, one by one, we will study. First one is the plankton. So, the group organism which is not being able to resist the movement and flow the water mass is called plankton or biocestal. Plankton can be differentiated as phytoplankton, zooplankton, protozoa plankton, viruses, and bacteria plankton also there. So these plankton, they don't have any specific type of mechanism of movement, and um, these plankton type of organisms, they can move when water current is able to move. So phytoplankton is there, and they are mainly microscopic algae and blue-green algae, and it is a varied community in terms of systematic and mainly composed of forms smaller than 500 micrometer. Sea phytoplankton are dominated by diatoms, dinophyta, whereas freshwater phytoplankton are dominated by the cryptophytes, diatoms, green algae, and blue-green algae. So different type of phytoplankton is there in the sea and in the freshwater. Freshwater phytoplankton is more diverse compared to sea. In the freshwater, cryptophyton, diatoms, green algae, and blue-green algae is there. Whereas if you will see the sea phytoplankton, diatoms, and dinophyta, it will be there. So the microflora, which is smaller than 50 micrometer, we are counting as a phytoplankton. It may be present in the sea, it may be present in the freshwater. Then zooplankton. They are the small water animal that occur in the plankton. There are three systematic groups that occur in freshwater: rotifers, brachiopods, and copypods. And the seawater plankton is composed of copypods, ctenophores, urochordate, and aeroworm, as well as some species of snail. Most of them are filtrators and or predators. So different type of zooplanktons is there, and they are small microscopic animals, organism, and they are divided in differently in freshwater like rotifers, brachiopods, and copypods. But seawater, if you will see, they are having more diversity. Copypods is there, and ctenophores, urochordata type of organism, arrowworm also there, and some small species of these snails also present. So they are the main diversity of zooplankton present in the seawater and the freshwater. Then protozoa plankton also is the part of the plankton, and they are also microscopic, consist of protozoa, and they are occupying the water zone like flagellates and ciliates. They are the main consumers of bacteria. Moreover, most ciliate, the organism which is having cilia, and these ciliate feed upon the flagellates, algae, and smaller ciliates. So the protozoa itself feed on the zooplanktons. So zooplankton, if you will see, they are the smaller type of microorganism compared to protozoa, and protozoa type of plankton is depend on the zooplankton. Okay, so next is the heterotrophic bacteria. Plankton is there, and they are occupying water which are abundant in organic compound, and the amount of bacteria in open water varies between 105 to 100 cells, 7 cells in per ml. So heterotrophic bacteria plankton is available only the water which is rich in organic type of compound. If suppose the water is contaminated by the sewage water, then it is very rich in the organic type of compound, and the bacteria population will increase because bacteria required organic type of compound to growth and development. That's why heterotrophic bacteria plankton will be available more. So it is a type of indicator when you are having the bacterial type of plankton in the water. So it means that the water is contaminated. If water is pure, then chances is very less that bacteria and fungi and viruses contamination is height, viral plankton, and they are smallest element of the plankton. Their numbers may be very high in various fresh and seawater habitats, and viruses are next to the protozoa, a crucial factor in bacteria motility. If your viruses is there, so that the viruses can able to infect various other type of microorganisms, and these viruses can able to reduce the like other microorganisms. They can able to control the microorganism population, because they will infect and they will able to kill these other type of uh, phytoplankton or plankton. Then periphyton. Periphyton occupy the shoreline zones, and they are a group of organisms that create outgrowth upon various objects and underwater plant. And most of the time they usually consist of small algae, diatoms, green algae, bacteria. And moreover, the various settles or semi-settled protozoa, eelworm, oligochaetes, insects, larvae, and even crustaceans make up the periphyton bioconsists. So basically, these periphytons are occupying the shoreline zones. They are not present in the where the main water in middle of the water source, but they are the present in the shoreline area. And the diatoms, green algae, bacteria is the main part. The small algae also is the part of the uh, periphyton. So periphyton has a characteristic complex bioconsensus, and many ecological relationships can be observed between its components. So these microorganisms, they are having ecologically very important, and they are balancing with the uh, higher organism to the lower organism, and they are very important for those organisms which are feed up on these type of microorganisms. Then benthos. Benthos are the those organism which is present in the bottom habitat and is occupied by a group organism we are calling benthos. So the organism which is living in the bottom habitat we are calling benthos. So the muddy bottom contain an abundance of organic compound, and that are created as a result of dead matter or decomposed, or when the these fallen part of the plants and animal, they will fall down in the muddy bottom, and these fallen part of the plant and animal, they will start decomposing, and when they will start decomposing, so the other microorganism, it will have opportunity to utilize those decomposed part, and they can get the energy from them. So at a great depth, the bottom is free from any plant which due to the lack of light. So these type of decomposed part of the plant and animal, it will be there. So however, the absence of oxygen support the development of among other uh, anaerobic type of microflora, because these microflora they are totally depend on these decomposed type of plants and animal parts. Among the benthos microflora, the most numerous are bacteria and fungi. They are the decomposer type of bacteria and fungi. And some animal like detriophages is there, and both of the above group are responsible of decomposition of the organic matter, and benthos of the shallow reservoir may also contain some type of algae. So these benthos, because in absence of light, they are totally depend on the decomposed parts of a plant and animal, and there is two type of organism is there, bacteria and fungi, and some small animals also there, and these small animals, they are all with the bacteria and fungi, they are dependent on the decomposed type of body, and they are getting energy when they will decompose this body parts. So here the four different type of groups is there. First one is Houston or Newstone. Second one is plankton. Third one is periphyton, and then benthos is there. So the the lowest part of the any water body which we are calling benthos is the benthos, and the Houston. This is the fluvium, the near the shallow water, and the plankton. There is the water which is moving through the water current, and the periphyton is there, which is near the shoreline of the water bodies. So these four type of organism it will be present in the any type of water bodies. Now sources and type of pollutant. So what type of source is there, and though these sources are related with the pollutant, and most of the pollutant are coming from these sources only. So domestic and industrial sewage, they are disposed of in the surface water, and even though nowadays the concentration of the disposal of water is increasing, so the groundwater also having contamination. So various compounds that affect the bioconsensus of water reservoir, and besides sewage pollution is also caused by rain runoff which wash away different fertilizer and crop protection product. So when the farmer is spraying different type of herbicide, pesticides, and the bactericides and fungicides, these different type of chemicals when the washed off with the rainwater or irrigation water, so this water can able to take this these type of chemicals, and they will be go inside the water bodies, and then they can able to reduce the contamination, chemical contamination. So moreover, the pollutant also transfer into the water from the surrounding air, and this usually result from the industrial dust which fall directly into the water or in the washed away from the ground surface by the rain. So important gases like sulfur dioxide, nitrogen oxide, carbon dioxide, and other dioxide which get into the water mainly, and these type of gases is the coming from the industrial area, and these industrial area is releasing the gases, water, and garbage, and this is the main source of the pollutant. Some of the above compounds undergo microbiological decomposition relatively easily and becoming food for heterotrophic microorganisms. So other are resistant to such decomposition and are harmful and toxic to the microorganism also. Example of these are the cyclic compound like engine oil, lubricant, chlorinated hydrocarbon, pesticide, and the mineral pollutant, heavy metals, salts. These are all they will not decompose because they are highly toxic to the organism also. So when these toxic type of compounds they will be disposed of directly in the potable water, so they will be persist in the water for longer duration and they will not degrade in the environment, and they are the main type of pollutant which will create problem for human life also, and other organism which is totally dependent and they are drinking these type of open source of water. So the pollutant, the main source, if you will see the two type of sources there, the sewage, which is household coming from the household, and the industrial garbage and sewage, which is directly dumped off in the open area. Now, organism which is part of the potable water, and when it is increasing the population, increasing beyond the level, then water is not potable. So viruses, protozoa, parasitic fungi is there, and beside pathogenic bacteria of surface water into the which municipal and industrial sewage is disposed of. So water also contains significant amount of pathogenic microorganisms such as poliovirus, and they are responsible for causing anemia disease, polio, and enterovirus which cause intestinal infection occur even in slightly polluted rivers. So these viruses, when they will be present in the drinking water, so they will cause problem with the spreading this type of virus disease. Protozoa infection of the digestive tract caused by the protozoa may come from the contaminated water, and most parasitic protozoa produce cysts which are able to survive inside their host in unfavorable conditions. When the condition improves, cysts transform into so-called trophozoites and the vegetative form occurring in the human. Then parasitic fungi is there and it is present in the polluted surface water. Parasitic fungi can also occur for example, microsporum species, trichophyton species, and epidermophyton species. They are dermatophytes, causing ringworm and other cutaneous infection when we will be in the contact of these type of fungi. So these viruses, protozoa and fungi, they are main causing agent of the different type of microbiological disease. And when the water is polluted and by the garbage and the sewage, so that these virus, protozoa and fungi, they will be contaminate the drinking water. And when we are utilizing without proper treatment or proper disinfectant, then these water can able to cause the infection. So the main thing is that whenever you are drinking or using drinking water, you have to disinfect it and then you can utilize it. Now, first organism is coliform and coliform type of bacteria, and this bacteria is different type of, we are dividing in different type of group like coliform group, and mainly made up of strain of E. coli as well as the genera of Enterobacter, Citrobacter and Klebsiella. They are detected on media containing lactose at 37 degrees centigrade. So these coliform bacteria we can able to detect using the lactose containing media. We will able to learn the method how we can identify coliform bacteria in the water. So the many groups is there in the coliform bacteria like Enterobacter, Citrobacter, Klebsiella, and different strains of the E. coli, and they are all combinedly we are calling coliform bacteria. So fecal coliform are mainly strain of E. coli and only some of the strain of Enterobacter, Citrobacter and Klebsiella, and they can able to ferment the lactose at 44 degrees centigrade. So the presence of coliform or fecal coliform in the water sample indicate the recent contamination of the with the fecal matters, sewage, soil, or with decaying plants. For most type of water and quantitative determination of both group of coliform is recommended. So we can able to quantify the how much coliform bacteria is there in the water, and if it is more than permissible limit, then the water is not potable. So when any natural source, if the coliform bacteria is increasing, that means that somewhere the sewage water and the soil or fecal matters is disposed of in the water bodies, and that's why the when these material will be disposed of in the water bodies, so the coliform bacteria population will be increased. So number of coliform bacteria we can count it, and we can able to decide that water is still potable or not. These tests we can able to utilize, and when we are doing, and these tests can indicate that whether water sample is having fecal infection or not. So when we are putting these water sample into the media which is having the lactose, if the bacteria can able to utilize the lactose, they can able to produce gas, and when gas will be produced, so that the our microtubes, it will be float on the media, and because they will, when they will utilize the lactose, they will produce the gas, and these gas filled tubes can able to float on the media, and when gas is produced, so that we can able to know that the fecal bodies is present in the water. Okay, if there is no gas, it means these coliform bacteria is not present, and coliform bacteria is not present, that this means no gas is there. Now we have to confirm the test whether what type of coliform bacteria is present. So for that, lactose probes tube we have to prepare, and the brilliant green lactose bile broth especially we will prepare for the identification of the coliform bacteria, and this medium inhibit the growth of the lactose fermenters other than coliform, thus gas formation in lactose bile growth. Green, green lactose bile growth medium constitute a confirmative test of coliform present. So this medium, brilliant green lactose bile growth, we can utilize for the confirmation of the coliform bacteria present in the water sample. Then other type of media like eosine, methylene, blue agar, EMB agar media is also we can utilize to streak on the bacteria sample. And when the coliform bacteria is present like coli aerogenes organism, and they can produce very characteristic type of colonies on the EMB media, and small dark, almost dark center with the greenish metallic sheen type of colonies. If it is present, then it's a confirmative test of the coliform bacteria. Then other completed tests also we can do when we are culturing these coliform samples. So lactose growth coliform produced by it will produce the gas, and gram staining also there. So coliform uh, type of the germ they can take negative staining, gram negative staining. So gram negative staining we can do when the coliform bacteria is present. Then bacteria native of natural water. Bacteria whose natural habitat is water are not well known, partially due to the fact that the many of them are difficult to grow in laboratory media. So the group include the following: higher bacteria, sulfur bacteria, or iron bacteria. Then second type, appendaged bacteria attached to some inanimate objects, and large spiral form bacteria. So these different type of bacteria native to natural water is present, and we can able to determine their population by microscopic analysis. Then fourth type, a variety of bacilli like pigmented form Serratia and Chromobacterium violacum, and non-pigmented form of Pseudomonas fluorescens, thermophils, aerobics, and anaerobic sopriforms. Then focus forms bacteria also present, and nitrogen fixing bacteria, nitrifying bacteria also is the part of bacteria community. Then bacterial contamination of natural water. Bacteria from air and soil. The number of bacteria in the air, and the close relation to the amount of larger suspended particles of dust, and which flow their way into the natural water through settle out or be swept down by the rain. And soil contamination contain tremendous number of bacteria, most of which are found in the upper 6 inches of the soil. So when the heavy storms is there, and soil erosion is there, and soil, we know the soil is very rich in the bacterial population. So when soil is erode through the air and the water, so it will carry the water microorganism population with them, and these microorganism population can able to contaminate the water source, because ultimately soil can easily stuck in the water source, and the microorganism present on the surface of the dust and soil, and these bacteria can able to cause the contamination of the water bodies. Now, others are present in contamination coming from excreta of human and animals, flesh of animals and human who have died of the infectious disease. So, if the human body or animal bodies, they people are throwing directly to the freshwater bodies. So these animals and human bodies can able to cause the various microorganism degradation, and these degradation when it will be there, so microorganism population will be increased very fast. So excreta of human or animal if directly releasing in the water bodies, so again the contamination also can be increased. Then direct contamination water by the human and animal excreta and normal intestinal flora which may be found in the water, and if it is present in the water or potable water, then we can directly indicate that the excreta of the human or animal directly releasing in the water source. Then large number of coliform bacteria, Clostridium perfringens, alpha hemolytic fecal streptococci or enterococci, these type of microorganism, it will be the part of the drinking water. If it is there, then it means that the human and animal excreta directly releasing in the water bodies. Pathogenic organism shed from feces of infected person and carriers includes the bacterial enteric pathogens, virus source. So if it is there in the water, then we can say the water is infecting from the pathogenic infected person, and they are directly without eating directly throwing the infected particle into the water bodies. So if the hospital waste and the infected person waste, if we release directly into the water bodies, so bacteria infected person can able to main cause of the infection in the water. Now bacteria found in surface water. So many type of bacteria, it will be the part of the surface water like aeromonas. These are all the main causative agent of the enteritis, and Campylobacter jejuni is there. Campylobacteriosis it will produce, and E. coli basically it will produce the urinary tract infection, neonatal meningitis, intestinal diseases, and Pseudomonas shigelloids is produce the Pseudomonas infection. Typhus is producing typhoid fever. Salmonella is producing the salmonellosis, and Streptococcus infection can able to produce the intestinal disease. Vibrio type of bacteria can able to produce the cholera, Vibrio cholera. So these type of bacteria can able to cause the different type of infection when they are present in the bacteria. If we will utilize as a drinking water, so these disease they will cause, or these type of infection can be caused by these type of bacteria. Now viral source of waterborne disease, hepatitis A, Norwalk type of virus, rotavirus, enterovirus or reovirus. If the contaminated water is present these type of virus, then it will be easily enter into the animal and the human, and they will cause different type of waterborne virus disease or viral disease. Now other indicator or organism, if they are present or if they are the part of the water, then they will cause some disease, and they are the indicator organism because they are having some specific characteristics. So one by one we will discuss these indicator organism and their characteristics and their significance. So first organism is Clostridium perfringens is there, and if you will see their characteristic, it is an anaerobic spores formers, gram positive, rod shaped, and exclusively of fecal origin. Spores are resistant and persist for longer period. And the significance, you can see that it is useful indicator of past pollution, a tracer of or less hardly indicator and protozoan and viruses. So if these Clostridium perfringens is present in the water sample, then it is an indicator of past pollution. Then bifidobacterium and bacteroids, if it is there, then it will show that the short time of therefore it's present such as the relatively recent type of pollution. Then Pseudomonas aeruginosa, if it is present in the water sample, then this organism is no value as indicator of fecal pollution. However, coliform do not show it as indicator of contamination of swimming pool water, as the contamination is not from the fecal origin. So Pseudomonas aeruginosa, if it is there in the water, then it directly not related with the fecal infection, then fecal type of contamination. It may be related with the some other type of source of contamination. Then Staphylococcus aureus and Candida albicans, if it is there in the water bodies, then it will indicate that the recreational water is contaminated with these type of infection. Then Aeromonas hydrophilia, and if it is present in the water and it is associated with the nutrition rich condition, it has been suggested as an indicator of nutrient rich status of the. Then factor affecting the kinds of number of bacteria. Types of water we'll see. First one is surface water. Surface water composed of stream, lake, shallow well water, and opportunities for contamination are mainly bacteria present in the water, and many type of microorganisms they can be disposed of with the garbage and other type of contaminated water. Water from the deep driven wells, water has undergone effective filtration in order to reach the surface strata, hence this only contain a few number of bacteria. So amount of organic matter present, more nutrient there is present in the form of organic matter. That's why the number of bacteria will be more in the when organic content will be more in the water. If temperature is not suitable for the growth of the bacteria, then the bacterial population will not be enough in the suppose if the water bodies is having high temperature, then the water contamination of the microorganism is low. Microbiological examination of water. When we will see in the microscope, and these microbiological examination will tell different type of contamination present in the bacteria. So numerous waterborne pathogens, individual pathogens, number may be too low to detect in a reasonable size of water sample. Isolation and detection of some pathogens can take several days and week of month, and we have to identify particular type of bacteria and their source. So it will take little bit longer time to identify the source of the contamination. Then absence of some particular pathogen does not rule out the presence of another type of bacteria. If particularly one type of bacteria is there, then we cannot say that other type of bacteria is not present. Water purification before the drinking purpose we have to do. So water purification form a critical link in promoting public health and the safety. It involves variety of steps which depend upon the type of impurities in the raw water source. The major operation done on sedimentation, flocculation, filtration, and disinfection. So these different type of methods we can utilize to disinfect the water, and sedimentation, flocculation, filtration, and disinfectant we can utilize to remove these contamination from the water. So first method is mechanical method. In mechanical methods, we can storage and filtration two methods we can utilize. First one is in the filtration, slow sand filtration is there, and second one is the coagulation and rapid sand filtration. Then chemical methods is there, second one, and the chemical methods we are utilizing for the large scale decontamination process of the potable water. Hypochlorite and liquid chlorine we are adding, and small scale we are adding the hypochlorite, UV light, and iodine in the household type of decontamination of potable virus. Now unit process and operation specific impurities removal. If the color and precipitate, then we can do the aeration, chemical oxidation, ion exchange chromatography, or column we can utilize, or sedimentation. The sedimentation we can use to remove this color and precipitation. Then if water is having calcium, magnesium, then we can use to chemical precipitation or ion exchange chromatography. Then if water is having turbidity, then we can use the dosing, mixing, and flocculation and settling type of filtration. And if water is having taste and odor, then we can use the aeration, chemical oxidation, and absorption. If we can have if we have the microbial infection, then we can use the radiation, ozonation, and chlorination, and we can easily remove these various type of microorganisms in the water. Now some useful references I will quote here. Handbook of Water and Wastewater Microbiology by Duncan Mara, Nigel J. Horn, is very useful handbook. Then water microbiology types, analysis and disease causing microorganism by and A, Lutsenko, Basil, Planktonic is there, and this is also another very useful. Now some important question. Which of the following is generally not referred to the sewage system? Option A sanitary sewers, option B storm sewers, option C combined sewers, option D is solid sewers. And next question, which of the following form of water is the most contaminated? Option A is underground water, option B is rainwater, option C is surface water, option D water stored in ice cave. The next question, alum is used for which of the following purification process? Option A is sedimentation, option D is filtration, option C is disinfection, option D ozonation. Anyone write the answer you have to choose. The next question, the coliform group of bacteria includes all the space bacilli. Option A is aerobic, option B is anaerobic, option C aerobic non-sporulating, option D is aerobic gram-negative non-sporulating bacteria. So anyone correct answer you have to choose. Which of the following organism does not belong to coliform group of bacteria? Option A is Salmonella, option B is Serratia, option B C Enterobacter, option D Proteus. Anyone correct answer you have to choose. Among the following, which microorganism act as a representative of fecal streptococci group? Option A is Streptococcus fecalis, option B is Streptococcus bovis, option C is Streptococcus equinis, option D is Streptococcus fecaci. So anyone correct answer here. Now see you in the next class and the next topic. Thank you.